Welcome to the first tutorial in bioinformatics on my channel. So in this tutorial, we will be using BLAST to identify homologous proteins. So what is the BLAST? First, we have to answer this question. It's actually a database, which means that the abbreviation stands for Basic Local Alignment Search Tool. So it's a kind of a database where we will be searching for the different sequences that different researchers have already deposited from around the globe. And if our sequences matches the sequences which are already in the database, then we get results or similarities, which means that our sequence shares some or all sequences which are already deposited into the database. So this is how we can identify if our organism whose sequence is deposited in the database has some common ancestor, because if two or more than two sequences share each other, it means that they have some common parent or common ancestor. And that is actually the concept of homologous proteins, which means that if different sequences of proteins matches each other, then they are homologous proteins. And it essentially means that their structure is similar, because of course if the sequences matches, which means the structure is similar. All right, so the first question is, find a record for hog1p protein. The accession number has been given to you, as you can see on the screen, in the gene bank database. Also the link of the database is given, which is ncbi. So provide answers to the following questions based on the information available for this protein in gene banks. So first question is, from what organism, species and strain does this sequence originate? All right, so to find the organism from where this sequence originate, we will just copy this link and paste it into our browser. Click enter. All right, so now we are with this interface in front of us. It might change because I'm recording in 2024. It means that it may change the look in the coming years. But the important thing is that you see here word protein, and also if you open the drop down list, you will see different parameters. But since we are interested in protein, so we'll be selecting protein, and then here what we will be doing, we'll be just writing our accession number. In this case, we are lucky that we have been given the accession number for this protein, so we just copy here and then hit enter or search. This is one way of looking the information that we are required to provide. So this is the name of the species, Saccharomyces cerevisiae, and this is the strain. What we will do, we will just take this information and paste here, since this is the required. All right, the second question is, on what chromosome is the corresponding gene located? To find this, we will go down and look if there is some information related to chromosome. So if you go or look under features and then source, you will come to know that chromosome 12 contains the information. And if you don't want to do this kind of long or a bit tedious job, you can just open a find function and just write chromosome, and then you will be given with highlight of chromosome, so you can easily write the answer, chromosome 12. So we will paste here that on chromosome 12 the corresponding gene is located. Okay, what is the biological function of this protein? To find the biological function, we go further down, and here in CDS, if we read the note, so from here till here, this is the function or functions of this protein. So we can just copy here, and later we can read while it let it paste like this. Okay, so it is actually mitogen activated protein kinase involved in osmoregulation, controls global reallocation of RNAP2 during osmotic shocks, etc. etc. What are the length and the molecular weight of the protein? As I suggested earlier, we can just write here our keyword if we can find something. So for this, we couldn't find. What about weight? Or how about if we just write WT? Yeah. So this is the molecular weight of this protein. We can just copy this and paste. This is the weight. And for finding the length, actually it's, if you go down here from 1 to 435, this is the length of the sequence. So we can say that 435 is length of the protein, and 48727 is the molecular weight of the protein. All right, now the last question of this exercise is, what conserved domains or regions does the protein have? Provide a description for at least one of them. To find the conserved region, if you go up and look on the right side of the screen, you will find identify conserved domain option. And also you can see here that it's written view conserved domains detected in this protein sequence using CD search. So if you simply click on here, as I have done it, you will be given or come up with this kind of a page. So here is the name of the conserved region. We can copy this for the sake of our exercise, and we can write STKC STY1 hog1 is conserved region, and the function is this description, catalytic domain, and the description states that it is catalytic domain of the serine. So this is how you find the information. This is one way. Another way is to find this kind of information is if you just click here on the left side on the NCBI, okay, and then you click on the right side on just on the blast. Since it is a protein, as you can see in the exercise, hog1 p protein, so we will be using this protein blast. And here if you just click and paste this number, accession number, since we can either supply accession number or gene IDs or FASTA sequence, we'll be getting the results. In our case, we have accession number, so I just supplied accession number. If you want to change the job title, when I paste it, it automatically pops up with this job name. But if you want, you can change whatever job name you want. For example, we can say since it was question one of our assignment, so we can say question one and component. What was the component number? um, 1 2 3 4 5 5. Well, since we need information for all of these questions, we can just leave it like this. Question number one. Okay, and then this is the important thing. You select this non-redundant protein sequence and no reference protein sequence, only non-redundant, because this option will give you all the sequences which are in the database and no need to change anything. Yes, if you need, it's better to click this option, so the original window of yours will remain here, because later you can, if you want for another protein, you can write the number here instead of going back and forth. If I will, for example, if I just click blast here, the operation will start in the same window, but if you will click show in the separate window or next window, and another window will pop up, so that would make our life easier. And here you can see the time. It means 3 p.m., but here from where I'm recording, it's actually 8 6 minutes past 8 a.m. So why this discrepancy? Actually, this time belongs to the server location. So if the server is in another location or in another country, then that's the reason the time is different. All right, so this is another way of finding the information that I showed you earlier. And here you can also see that this information or this kind of research has given us a lot of significant alignments in contrast to the previous approach that I used. If you go back to the start of the video, you will find how I searched. So, but anyway, and here you can see the percent identity 199. This, if you want, you can also click on this and it will just give you the order in descending. But here you can see that the first three results are 100% identical. But another thing to note is that the query cover for the second and third uh, result is 93 and 80%. So therefore, our best bet is to select a result which is 100% covering our query. That means that it has 100% matches from the database, from inside the database. And you can see the score is also highest, 910, as compared to the second and third result. So that's why we will just click on this. Either you can click here or you can click here on accession and a new window will pop up. So again, we are on the same page where we were before at the start of this video, because before I just put my number here. I can show you again to sum up. So if you in the start of the video, I have just put this accession number here and I clicked search, and it came with the same page that I just reach by clicking here. So that is how you find the information of homologous proteins in BLAST. I hope you have learned something useful for this exercise. And if you want some more information, you can actually find here, for example, instead of copying the name of organism and strain from here, you can also If you scroll down, you can find the name of the organism and their kingdom, genus, and species. And you can also find reference authors who has deposited or reported the sequences. And if you go further down to features, you can find length of the protein, chromosome number, the regions, and if you go further down in CDS, you can find the name of the protein or the gene, and then locus. And if you go down to the note, you will find different uh, description for the function of this protein or gene. So this is how you find information in BLAST. I hope you've learned something useful. Thank you very much for watching this tutorial. Please keep subscribing and liking my videos. Bye bye.